lecture will be on the biochemistry of vision. And the learning objectives for this lecture is we're going to talk about metabolism in several different parts of the eye. So what is metabolism in the cornea like, and how does the cornea protect itself from oxidative stress? Secondly, we're going to talk about metabolism in the lens and the basic biochemistry of cataract. Third, we'll be talking about metabolism in the retina, including what photoreceptors and photopigments can we find in the retina, what are the importance of retinoids in vision, and what's the biochemical mechanism of visual transduction itself, and a little bit about color vision and also the basics of color blindness. So the reason why it's really important to talk about the biochemistry of vision is basically the reason why we can actually see with the eye is because the eye allows us to see by converting light stimuli into neuronal impulses, and this is a largely biochemical process in which each structure of the eye has its own important function. For example, the cornea and the lens must maintain clarity at all times. The vitreous chamber and humor maintains isosmotic pressure and provides nourishment for the lens and the cornea of the eyes. And finally, one of the most important components is the retina, which contains the actual visual sensing apparatus that we will need to convert this photon stimuli into a neuronal impulse. And finally, one more thing is that because the eye itself is an extension of this nervous system, then major metabolic fuel used in the eye is also glucose, just like the nervous system. Now, let's start by talking about metabolism in the cornea. So the first question is, how exactly does the cornea stay clear? As we've talked about before, the cornea must always stay clear so that it can allow the diffraction of light. Now, it stays clear by two mechanisms primarily. First of all, it has to maintain an avascular state, which means no blood vessels are allowed in the cornea. And this is largely maintained by a protein called VEGFR, or vascular endothelial growth factor receptor, which functions to prevent angiogenesis. So here, as you can see, with the presence of VEGFR, what happens is that it can actually prevent the formation of blood vessels in the cornea. Whereas if we don't have VEGFR, then this can lead to the formation of blood vessels, or the process of angiogenesis. Now, the second mechanism is that it uses an ATP-driven water pump to control the water content and clarity in the cornea. Now, because it's ATP-driven, we then ask the question, how does the cornea obtain ATP from? So basically, the cornea generates ATP through glucose metabolism using two main processes, which is glycolysis and hexose monophosphate pathway. Glycolysis itself composes 30% of the glucose metabolism process in the cornea, while the HMP process makes up the majority of metabolism in the cornea. So 60% of corneal metabolism is performed through the glucose HMP pathway. And if you recall, the HMP pathway is a type of pathway that actually produces NADPH, which will play a very important role later on. Now, as you know, that the cornea is the most outer part of the eye, and it's constantly exposed to environmental factors, including oxygen, meaning that the cornea is prone to the presence of reactive oxygen species, or ROS. Now, the problem with ROS is it can actually cause a lot of damage to tissues through processes such as lipid peroxidation and also through protein oxidation. Now, this is where NADPH plays an important role. So the cornea actually can protect itself by neutralizing these ROSs through glutathione, but glutathione itself needs a lot of NADPH in order to perform optimally. So the question is, how does NADPH enhance GSH? GSH is glutathione, in order to neutralize ROS. So basically, how does NADPH work? Well, it increases glutathione production. You see, with each presence of ROS, GSA actually takes the blow and it gets oxidized into an oxidized form of glutathione. Now, when we actually need more GSH or glutathione, then what happens is that the NADPH from our HMP pathway before will convert the oxidized glutathione into reduced glutathione, therefore providing us with more glutathione that can be used to counteract these ROSs. So now that we've gotten the cornea out of the way, we'll move on to metabolism in the lens. So the first question is, how does the lens stay clear? Because just like the cornea, it needs to maintain a clear crystalline state. Well, just like the cornea, it also maintains its osmotic balance through a pump, and it's also avascular. And that's the reason for why the lens is able to stay clear in avascular is because the main component of the lens is just water and also proteins, specifically alpha, beta, and gamma crystalline proteins, which is synthesized by the outer epithelial layer. Therefore, this leads us to the next question. So how does the lens generate ATP? Now, a healthy lens actually also uses glucose metabolism, but in a healthy lens metabolism, we dominantly use anaerobic glycolysis to metabolize glucose, and some of the glucose is also metabolized through the TCA cycle aerobically, and a small percentage is metabolized through the hexose monophosphate pathway. However, what you need to really know is that glucose metabolism in the lens can also activate what we know is the polyol pathway, which is a pathway that occurs when there is an excess glucose state, meaning there's too much glucose in the lens. And this is the basis of a lot of some diseases such as cataract. Now, you may ask, what is the polyol pathway and how is it activated? Well, the polyol pathway itself is actually a very physiologically important pathway. Basically, what it does is it converts glucose to fructose, and this plays an important role in a lot of cells, such as sperm cells. However, in the lens, this is not actually something that we want. In the lens, the polyol pathway is only activated in conditions of excess glucose, such as a person with diabetes. So what happens is that the glucose in the lens will be converted first into sorbitol. This requires the work of an enzyme called aldose reductase. And the sorbitol is then converted into fructose by polyol dehydrogenase. Now, this is the basis of a disease that we know as diabetic cataract. So how does the polyol pathway cause diabetic cataract? Well, first of all, you need to know that cataract is a disorder of lens opacity. Although there is different kinds of cataract, when we talk about diabetic cataract, this is caused by a loss of osmolarity control due to an increase in sorbitol. So the reason why there's a lot of sorbitol accumulation in the lens is because when we have a lot of glucose in the lens, then a lot of it will be converted into sorbitol. However, we don't have enough polyol dehydrogenase in the lens to convert this into fructose. So this sorbitol will just accumulate and accumulate, therefore causing osmotic imbalance in the lens. Now, eventually this is going to cause lens damage, which we also know as diabetic cataract. So that's the mechanism behind diabetic cataract, is because of the activation of the polyol pathway that leads to sorbitol accumulation and lens damage. So now we move on to metabolism in the retina. So there's going to be some subtopics when we talk about metabolism in the retina, such as what's the main function of retina, how does it generate ATP, and then what cells do we use for visual transduction, and what pigment is present in each photoreceptor cell. And then finally, we'll talk more about visual transduction process. So we know that the lens and the cornea has a main function of staying clear in order to diffract light, whereas the retina itself has a main function of visual transduction. So this is the light entering the eye. Once it crosses the clear cornea and the clear lens, then it will be diffracted and hit the retina. And this is, as you can see, this is a physical process. We have light entering the eye. Now, what happens is that this physical process needs to be turned into a neuronal impulse, and this is largely a biochemical and an electrical process. In the retina, we have two different photoreceptor cells, which is the rod and the cone cells, and this is connected to bipolar cells. So these bipolar cells are actually the ones that's going to convert this into a neuronal impulse, transfer this to the ganglion cells and into the optic nerve. So rod and cone cells, bipolar cells, ganglion cells. Now, in order to perform this function, the retina, of course, needs energy, and the way they generate ATP is also through anaerobic glycolysis, just like the lens. We talked about photoreceptor cells. There are rods and cones. So this is a section of the retina. As you can see, this is an electron microscope image. So in regards to rod cells, there are around 200 million rod cells in our retina, and cone cells are much smaller in amounts relative to the rod cells. There's only 6 to 7 million cone cells in humans, and rod cells are responsible for vision in low light, while cone cells are responsible for color vision. So this is very nice electron microscopy of the rod and cone cells. As you can see, there's so much more rod cells compared to the green cone cells. Now, inside these rod and cone cells, we have photopigments, and these are the ones that's going to convert the physical light, photons, into a biochemical reaction. So when we may ask, what exactly is a photoreceptor pigment? So a photopigment is basically just a 7-transmembrane receptor, or a serpentine receptor. If you recall, when we talk about cell membrane and signaling, a 7-TM receptor or a serpentine receptor is usually coupled with a G protein. So in this case, this is the retinal membrane. This is our photopigment. This photopigment is in red, and it's called an opsin. Opsin is the 7-TM receptor that we can find in the retina. Inside the opsin, we have this blue component, as you can see, that is a prosthetic group that we know as 11-cis retinal. 11-cis retinal is a prosthetic group that's always bound to the opsin protein in red. So they're bound together and they're transmembrane, and they're bound to a G protein. Here, the G protein in the eye is called transducin. Now, one of the most important parts of this complex is exactly this 11-cis retinal, because this is the one that's going to undergo changes or conformation when it is hit with a photon of light. Now, what we need to take note of is that photopigments or opsins in the retina cells are different depending on what kind of cell it is. If it is a cone cell, then the pigment is called an iodopsin, and if it is a rod cell, then the pigment is called rhodopsin. All these opsins have the prosthetic group that we call 11-cis retinal. The only difference is the opsin component. For example, in cone cells, which is responsible for color differentiation, the blue pigment contains 11-cis retinal group and a blue opsin protein. The red pigments have an 11-cis retinal prosthetic group and a red opsin protein, while the green pigment has an 11-cis retinal and a green opsin. So that's where the difference lies. So 11-cis retinal is a prosthetic group that is found in all kinds of opsins, no matter what the kind of cell is. So yes, that's what a photoreceptor pigment is. And as you know, we talked about how the opsin contains 11-cis retinal. If you recall the vitamin and mineral lecture, you'll know that these retinals come from a compound called beta-carotenes in our diet. So when you eat diet rich in beta-carotenes and other provitamin A carotenoids, what happens is that in the intestines, these will be cleaved into two all trans-retinol molecules, and this will be isomerized into 11-cis retinol in the retina, and this will then be oxidized into 11-cis retinal. And then once it's in the form of 11-cis retinal, then this group will attach itself into an opsin protein by protonating a Schiff base. Now, the reason why we need to recall this fact is because beta carotene is actually present in a lot of different kinds of foods, and therefore vitamin A deficiency due to dietary problems is usually one of the most important and preventable causes of blindness. Now we're going to talk about how does the retina convert the photon light into a biochemical signal. And this is a process called phototransduction. And before we go into the biochemical mechanism, maybe we first need to remember a few principles just so that we don't get things mixed up. First of all, what you need to know is that there's a molecule called cGMP whose job is to keep this ion channel in the cells open. Now what happens is that open gates equals depolarization, and depolarization will lead to the release of glutamate, which causes inhibition of the bipolar neurons. So this is the bipolar cells, and these are the rod cells. Remember that cGMP keeps the channels open, and when these channels open, what happens is that depolarization will occur, as in this case. So this is in the dark. The cGMP is plentiful, and therefore it actually keeps the gates open, leading to depolarization and the release of glutamate. Glutamate will actually inhibit bipolar cells, so it's got a negative effect towards bipolar cells. On the other hand, when in the presence of light, what happens is that there is going to be less cGMP, therefore the gates will be closed and the cells will be hyperpolarized. Therefore, no glutamate will be released here, and there will be no inhibition towards these postsynaptic neurons. So this is the basic principle. The cGMP keeps the gates open. Open gates creates depolarization, and depolarization causes the release of glutamate, which inhibits bipolar neurons. Now that you know the principles, we can talk about the mechanism of phototransduction. So when light comes in through the eye, it will hit this 11-cis retinal. This is the 11-cis retinal. It's in a bent form, and it will convert it into a linear isomer called all trans-retinal. This will initiate the next process. So once 11-cis retinal is converted into all trans-retinal, this will initiate the next process, which is to activate the rhodopsin. Activated rhodopsin, which will then activate transducin, which is the G protein. When the transducin is activated, it will exchange GDP for GTP, and the GTP will then activate the phosphodiesterase units. The phosphodiesterase units will create hydrolyzation of cGMP. So once cGMP is hydrolyzed, there will be no more cGMP to open the gate. So phosphodiesterase breaks down cGMP. There's no more cGMP left. Therefore, the gates are closed. That when the gates are closed, then we know that this will actually cause hyperpolarization of the membrane, therefore inhibiting glutamate release and also activating the postsynaptic neurons. So that's actually what we just talked about is phototransduction basics in light vision, but in color vision it's actually very similar. The only difference is that we have 3 different kinds of photopigments in the cone cells, and they're different because of their sequence of the amino acid sequences. The light sensitive component is still 11 cis retinal, remember, but the absorption properties of the retinal is modulated by the type of opsin. So this is an opsin. As you can see, this is an example of the blue opsin. The open circles here show the amino acids that are the same as rhodopsin, and the colored circles are actually the amino acids that are different to rhodopsin. So the reason why they can see different colors is because we can absorb different colors, mainly due because of the amino acid sequences are different between these opsins. Now, as you can see, the blue opsins absorb these wavelengths, the short wavelengths. So this is the blue spectra, and the green sensitive opsins absorb the medium wavelengths or the green spectra, and the red opsins absorb the long wavelengths or the red spectra. So the reason why, once again, that they can absorb the different wavelengths is because they have different amino acid compositions. So that's the mechanism of color vision. Now, finally, this protein, as you can see, these different kinds of opsins are, of course, encoded by our genes, and this is actually the basics of color vision deficiency. The blue opsins are encoded by OPN1SW genes, which is located on chromosome 7. It's autosomal. While the green and red opsin genes, so the OPN1MW and the OPN1LW, are both found on the X chromosome. So that is why color blindness, if you see in the community, is actually commonly an X-linked recessive phenomenon. So in males, for example, you either have a normal color vision male or a colorblind male, where in females you could be normal, colorblind, or a carrier. As an example, if this is a Punnett square between a colorblind female and a normal vision male, the female children will be carriers but not colorblind, while the male children will be colorblind. So that's the reason for why most cases of colorblindness is X-linked recessive and affects males more than females. So that's just a little introduction to the biochemistry of vision. So there's a couple of points, take-home messages that you need to understand. The firstly is that the lens and the cornea both have a job of maintaining clarity using the ATP-dependent pumps, and ATP in these two structures are generated mainly through glucose metabolism. In the cornea, this is mostly through the HMP pathway, while in the lens, this is mostly through anaerobic glycolysis. The reason why the HMP pathway plays a big role in the cornea is because we need NADPH for glutathione to work properly. And in the lens, one thing that you need to know is the polyol pathway, which can lead to cataract, diabetic cataract due to sorbitol accumulation. And then when we talk about the retina, what you need to know is the process of phototransduction. We talked about the differences between rhodopsin and iodopsin and the mechanism behind phototransduction. We also talked about a dietary vitamin A, why this is crucial for the function of retina, mainly because it is crucial for the production of the 11 cis retinal that is present in all of our opsins. And we now also know that color vision is regulated by the absorption spectra of each opsin, and this is determined by the amino acid sequences of the opsins. And all these opsins are still bound to 11 cis retinal. And the final thing that we discussed is that most cases of color blindness is an X-linked recessive trait and is most commonly found in males. These are some of the references that you may wish to read on just to expand on this material. Thank you for your attention.